Hi, my name is KIANNA, and today I want to talk to you about bats. So bats are emerging as an important animal model in studying brain development, neurobiology, and also aging. Why do they make such good models? Well, they live for a very long time in captivity, over 10 years. And their brains resemble that of non-human primates and humans a lot more closely than model organisms like mice or rats do. So this summer, I worked with a lab that studies Corolla perspicillata, otherwise known as the fruit bat, and I was looking at changes in the brain with aging. One of my projects was to take the brains of 2 to 3 month-old animals just after they're weaned by their mothers and perform immunohistochemistry on them. I found a novel population of entorhinal cortex cells that are positive for calretinin that appear in young bats but not old ones. So the entorhinal cortex. This is a region that is critical for navigational behavior, like going and finding your house or a source of food. And it's also the first region in the brain to be impacted by Alzheimer's disease in humans. Here I'm showing 2 sagittal sections of a brain, and they're both stained with calretinin. Calretinin is a calcium binding protein that is expressed in inhibitory neurons that are vital for neurological development. And so on the left-hand side, you can see previous work done at our lab by Dr. Timothy Morello, and he is showing the calretinin expression, which is shown as the red staining areas in an old animal greater than 5 years old. We assess this by body size and wing appearance. This is the hippocampus region here, and you can see that there are red cells. But in this white box, which is the entorhinal cortex, you can see a distinct lack of red staining in this area. Now compare this to an image that I took this summer. This is a young bat brain, and I've also shown here the entorhinal cortex. But you can see this amazing staining of red cells here. These are calretinin positive neurons, and you can even see their processes extending to the surface of the brain if you look closely here. So, to our knowledge, this is the first time that these have ever been detected. And our current working hypothesis is that these calretinin positive neurons are vital for organizing the entorhinal cortex during postnatal development. So our next steps, what do they look like? Well, we need to take time points between old and young to dive deeper into what exactly these changes look like over time. And I think that this is a really important reminder that the study of aging can't just include older age and end of life, but it also has to include the beginning of life too, and every stage in between. Thank you very much for your time.